All right. So, as I was saying, we're getting to a spot where we've covered our basic cell biology, and now we're going to be doing our different diseases of the week and see where that goes. So, everybody cough and worry that we have some kind of upper respiratory tract bacterial disease. And one of the things to keep in mind is that all these steps, you got bacteria in your upper respiratory tract all the time. It is only when the numbers start to get out of control that you have problems. Occasionally, if somebody were to blow anthrax into your face, you might inhale something that is just flat-ass nasty, but that's a good bit more rare. More commonly, people will be weakened immunologically, and the bacteria in the respiratory tract starts to get a foothold, and that can be pneumonia. And so what we're going to look at is the entry point, and additionally how the bacteria is being transmitted as well, because a good infection, if you want to think of it that way, is one that is going to get into you through some sort of opening and then begin replicating in a way that it can be expelled and spread. So, one of the first upper respiratory bacteria to talk about is Streptococcus pyogenes. And this is responsible for strep throat. How many of you have had strep throat? I don't think if I had, no one cared. At least in the modern era, if you have strep throat, they take that swab and they swab gagola in the back of your mouth, and then they're doing an antibody test where they have essentially looks like a pregnancy test kit, where they put this in solution, take a drop of the solution, put it on a test strip, and they look to see if you are carrying the bacteria, and at a sufficient low that it's detectable. And that's how they say, yep, you got strep throat, or no, you got something else. You have, a viral infection is causing inflammation of the upper respiratory tract. Then they give you your Z-Pack and send you on your way. So, strep is a streptococcal bacterium, which, if you remember, that means it's a chain. And it is gram-positive. It's got a pretty hard candy coating, a capsule on it. It is secreting an exotoxin. It is not motile, and then it also has a variety of proteins that, if the infection continues, can lead to a variety of different inflammatory problems, rheumatic fever, and this results, some of these are terms that have been used for a very long time when people talk about rheumatic fever. This is a bacterial infection, a strep infection, that is not just going into the lungs, but it's actually starting to become endemic and getting into the heart. And it can have pretty serious effects on joints. It can damage the heart. It's a pretty serious thing, which is one of the reasons why you never want to leave a strep throat or a strep infection necessarily run its course. So, it's possible for it to get and spread around. So, scarlet fever is a result of a strep infection. It is an immunological response to it, and it can result, because of the toxin, in a variety of things like being blind and going deaf. And Helen Keller, very possibly, through scarlet fever, had this. And a lot of kids, we're probably to a spot now where it's your great-grandparents, would talk about either people having scarlet fever or going through it. This is a bacterial infection that has actually gone systemic. There is, lovely, necrotizing fasciitis is another way that strep can actually manifest itself. Toxic shock syndrome also is something that strep is responsible for. Anything that produces different kinds of toxins, and so like the exotoxin, if you get enough bacterial load, even though you get on antibiotics and knock the bacteria back, you still have all this exotoxin floating around, and it can cause some fairly serious problems, things like toxic shock. Jim Henson, just as a little point here, he was not one to go see doctors much. I'm not sure if it was a religious issue or he just was not interested in going to see doctors. He had a bacterial infection that's probably started out as a strep throat type infection, let it go too long, and ended up having such an accumulation of bacteria, the toxin, that he died from it. All right. So another classic upper respiratory infection is a bacteria that causes diphtheria. Now, in the modern world here, we have all had our diphtheria vaccinations. This is generating immunological resistance against the bacterium, Corynebacterium diphtheriae. And it's not so much of a problem in the U.S. So it's gram-positive. It is got a club shape to it. So you can see this little bulbous end. One of the things this means is that in a culture, it's pretty easy to recognize it simply from the microscope. You don't have to do a whole lot of metabolic tests. It causes a variety of tonsil, laryngeal, various sorts of inflammation in the upper respiratory tract. Because of the inflammation, it results in lymph nodes enlarging in the neck, and that is referred to as this bull neck phenomenon or diagnosis. You get a really raspy cough with breathing difficulty. It used to be referred to as croup, and just sort of the hacking sound. And babies and little kids would get this, and it's not very pleasant. The exotoxin is actually produced from a viral bacteriophage that the bacteria has incorporated into its genome. So it's a danger in that in the membranes of the upper respiratory tract, it can be a problem. If it becomes systemic, it's a real problem. And that's really going to be one of the themes, is that there is going to be bacteria that have a specific entry point, and that presents certain conditions. If you let it go, you end up with a systemic infection, and that's pretty much always worse. So prior to the vaccine, there were millions of cases a year. It's now a fairly rare kind of condition because we've got a pretty good vaccine against it. So we mentioned here this idea of what's called an exotoxin. And these are proteins or glycoproteins, or sometimes they're just sugars, that, and they also be actually lipopolysaccharide stuff. So a combination of all these different biopolymers. And they have specific toxic effects. You undoubtedly know of botulism toxin, of Botox. Botox is able to interact with neurons in the neuromuscular junction and prevent nervous system signals from contracting muscle cells. And so you inject it, and all of a sudden the muscles that are contracting your forehead no longer have a neuronal signal, and they relax. So these toxins are produced by bacteria, and very often this is a really nasty part of it. And specifically, the exotoxin is an enzyme that is able to block protein synthesis, and that makes it a really potent and toxic metabolic poison. If you can't produce proteins, eventually the cell is going to have some problems. And it is, in fact, so potent, that one toxin molecule is going to kill one cell. So there are some really nasty things out there, and this is one of them. So the protein complex gets into the cell by something called receptor-mediated endocytosis, which, if you break that term apart, you have a receptor on the cell surface that binds to the protein complex. And that causes that portion of the membrane to be engulfed into a membrane, into a vesicle. And so this is known as endocytosis. And once you let this thing into the cell, it's actually able to release a component inside the cytoplasm that can now begin blocking the ability to make peptide bonds. And so it's a fairly stealthy and nasty kind of toxin that gets in fairly easily, and it doesn't take a whole lot to start screwing the cells up. So here is another kind of inhaled bacterium that are a class that we can begin kind of going through. Neisseria meningitidis is a gram-negative guy, and this gets in through the nose, gets into the sinuses, and it actually can begin working its way up into the brain. It's one of the things that you're often cautioned about picking at your face, popping zits in this sort of triangle, because it's right around the sinuses, and you can get a bacterial infection going in there, and it's possible to get it up in your brain. It doesn't happen a whole lot, but when it does, let's just think about that. Having a brain bacterial infection is way, way bad news. Meningitis is a general term for inflammation of the meninges, of the brain lining. One of the issues is the so-called blood-brain barrier. Essentially, there is systems in place that keep certain metabolites and a variety of different things from getting up into the brain. We try and keep the brain in a reasonably safe spot. It's not easy to get antibiotics up into the brain. And so it's a very difficult spot to kill bacteria. Or another way of saying this is bacteria can hide out in the brain and evade a lot of attack. So you pretty much, at this point, are going to be hoping that your immune system can knock it back. So the death rate depends on a lot on age, the kind of bacteria, the bacteria strain, and how quickly you're able to get treatment. So Neisseria is a gram-negative, aerobic, and not motile, but it can make its way around. Let's see, a couple of other guys that we wanted to mention as well. Streptococcus pneumoniae. This is the standard kind of bacterium that causes bacterial pneumonia. And again, what we have is these terms that are really very old, like pneumonia, and they're generically applied. So if people have fluid in the lungs, they've got difficulty breathing, and it's not due to like congestive heart failure, it might be referred to as a pneumonia. And so you can have a viral pneumonia, or you can have a bacterial pneumonia. And strep pneumoniae is one of the main causes of bacterial pneumonia. Another bacterium that perhaps you've heard of, Haemophilus influenzae, is a gram-negative. It is a anaerobic bacterium, and this can cause a variety of different kinds of upper respiratory infections as well. We have the Hib vaccine, and that knocks it back pretty well. One of the things to think about, and is why we talk about these different kinds of growth habits for bacteria, is you can get bacteria that can travel up into your sinuses and get pretty far up in there. And the sinus is, a fairly small cavity, and actually begin to build up fluid and mucus as the body tries to fight it, and you can generate an anaerobic environment up in your sinus. Most of the time, your sinus is open and is reasonably aerobic. When it becomes anaerobic, you actually are creating the conditions in which these different bacteria can now thrive. So we might be exposed to Haemophilus influenzae all the time. And it's one of the things to remember. This is not viral influenza. This is a bacterial infection, just with the name influenzae. And as long as you don't have the conditions for bacterial growth, you really are not too much of a risk. But as your sinuses start to clog up, allergies can start to do this, and the bacteria get a foothold, you start to generate a bacterial infection up into your sinuses that can become a chronic infection. And sometimes one of the treatments will be to take essentially a little crunchy drill bit and to go up into the nose and start kind of removing the gunk. And it's a little bit crunchy because there's actually cartilaginous material and who knows what stuff you shoved up your nose that you're busting apart and essentially bringing oxygen into this anaerobic spot. When you do that, A, you can breathe a lot better, and B, you have now generated an environment in the sinuses that is not conducive to this bacterial infection. And that is a standard kind of nasal treatment that people will do. Another thing that happens is you get nasal polyps, little growths in the lining of the nasal mucosa, can start to block off some of the passages up into the sinuses, and again start making things go anaerobic. It's one of these ideas that if you have parts of the body that are supposed to be aerobic, you have a lot of airflow moving around them, shifting them to anaerobic is a great way to start screwing yourself up. And the same is true for other parts of the body that tend to be anaerobic. If you shift the conditions, you can start screwing things up as well. So a couple more bacteria here. All right, so there are a range of different kinds of bacteria that can also cause upper respiratory infections. And you don't need to really remember the list exactly, but just be familiar. Pseudomonas aeruginosa, strep pneumoniae, Haemophilus influenzae. We've been looking at some of these. Some of these have more specific kinds of targets. So like the epiglottis in the back of the throat is one specific target. It can actually begin to swell and cause breathing difficulties. Presumably it's what killed George Washington. I don't know that for a fact, but it's a Wikipedia point. You can get middle ear infections from this. Sinus infections often result from some of these different bacteria. Tonsillitis, many of you have had this. It used to be a standard procedure to have your tonsils removed. Anybody have their tonsils removed? I still have all of mine. And it is a way of removing the lymph nodes that can be actually responsible for draining and pulling in a variety of different kinds of bacteria from bacterial infections. They swell up, they give you a sore throat. Removing the tonsils is not something done very often because really it's actually a fairly good defense mechanism. And what you probably should be doing is looking at the secondary symptoms or the primary symptoms that are causing tonsillitis. Laryngitis, pharyngitis, bacterial infections can cause this as well. So we can distinguish between the upper respiratory tract, and now it's going to be a good bit nastier, and that's the lower respiratory tract. So a couple of diseases perhaps you've heard of. Whooping cough. This is another bacterial disease, and happily we've got a really good vaccine against this. So it's a gram-negative. It's aerobic, which makes good sense because it's in the lungs. The exotoxin is paralyzing the cilia. So again, we have an exotoxin being secreted by the bacteria that's having a specific effect. And what this is going to do is prevent the cilia from flushing and moving stuff, the dust and debris out of your lungs. And I don't know if you had AMP or when you get into AMP, you'll know that the lungs are multiple branches, and you start branching off into tinier and tinier spots until you actually get to areas in the alveoli where you've got a single cell thickness between the air and the blood. And this is a very narrow passage, and you have to constantly be sweeping away dust and particles and goobers and snot and mucus to keep that passage open. If you lose the ability to move your cilia, you're going to begin to accumulate material and start having a fairly violent cough. Ugh. So the inhalation is that sort of whooping sound. It can last for a long time because not only you have to get rid of the bacteria, but then you also have the effects of the exotoxin as well. And we now prevent this with the diphtheria, pertussis, tetanus, what's the T? Regardless, the DPT, or now the DTaP vaccine. The exotoxin is a little bit different than the exotoxin that we were looking at with previously, but it has the ability to actually affect parts of the cytoskeleton that are involved in the waving of the cilia. If you look at the effectiveness of the vaccine, pretty remarkable. So we have lots of cases of whooping cough, pertussis, and it was a serious killer of infants. Vaccination efforts began in the 40s, and wow, we get it down now to a very low level. Now, one of the things that's interesting to think about is that this bacteria is probably still floating around all the time. So maintaining a vaccine is not a matter of herd immunity anymore. Maintaining the vaccine, in this case, the bacteria is present all the time, so you don't get herd immunity in that it's going to be around. You have to always maintain this vaccine, otherwise you start to have an increase. Now, I'm not sure that we know what this bump in 04 is about. It did go back down. So I suppose there's a possibility of developing bacterial strains that are not being recognized by the vaccine, but I would suspect that's not really the case. Nevertheless, it was a fairly small bump compared to what it's been. Okay, another bacterium that you've heard of, potentially, tuberculosis. And, again, we are reflecting on the fact that these are diseases that have been plaguing mankind for as long as we've been around. And so the terminology and the treatments, many of them predate the idea of cell theory and the notion of actual microbiologicals causing illness. And so there's a lot of things that float around about how you treat and how tuberculosis manifests itself. So TB, tuberculosis, it's an aerobic bacterium, and it is generally a rod shape. It's gram-positive, and it is a little strange for its cell wall shape. You end up with a bloody cough, fever, night sweats. Over time, it begins to affect the overall metabolism, and you end up with weight loss and a fairly high death rate if it's untreated. So, antibiotic treatment works fairly well for tuberculosis. The treatment, though, for tuberculosis is antibiotics for literally months, 6 months on antibiotic. And there is increasing amount of antibiotic resistance, which is a real serious problem. In New York City, there are antibiotic-resistant tuberculosis cases becoming more increasing in their frequency. And this is a problem because, what you do with antibiotic resistance is, all right, if this one doesn't work, you kind of go to the next level of antibiotic. And, as we've been talking about, antibiotic resistance isn't an if it'll happen, it's just a matter of when. And the activation of these specialized antibiotics is going to start inducing resistance to them or generating populations that are resistant. And so they're used very sparingly. So tuberculosis used to be called consumption because the body begins to waste away. X-rays reveal these essentially masses of bacterial infection and fluid that were known as tubercles. You can begin to hear them actually as crackles in the lungs and things. And so it's a pretty serious problem. Now, you may or may not have ever had a TB test. And this is done because tuberculosis still is in the population. And particularly with the elderly, it's not good to expose them to this bacterium, or little kids. And so for a TB test, what they will do is jab you with a little bit of either a killed or portion of the tuberculosis bacterium. They jab it in your arm, and they look to see if you have a response to it, if you have an immunological response. So if you've had TB before, you'll definitely have a positive TB test. One of the things with the TB test is that they jab you, and generally you're going to get a patch of red and a swelling. The amount of swelling and red that is a positive is really significant. So if you ever have one, you go, oh my God, I think I have TB. Nah, the positive is like really, really serious. So it is a fairly standard kind of test, and one that's done with healthcare workers all the time. So this is the progression of it, and happily, you can be exposed to it, and most of the time, not going to have any kind of infection. So this is often the case with lots of bacteria. You're exposed to it, and it just doesn't get enough of a foothold to bother you. Your immune response is good enough. You didn't get a big enough dose. And so a lot of folks can get it or can be exposed to it and not have any kind of specific effects. So the primary infection is going to be in the lungs. The bronchi spread apart, these trees, and they allow the tuberculosis to get in. It's being spread by coughing. So, in the middle of a reading about antibiotic resistance tuberculosis, and then you get on the L in Chicago, and there's some kind of janky-looking dude coughing on you. It's like, oh, great. Thanks a lot, sir. So, masks. Anyway, most folks end up with a latent infection. And this is an infection that is low level. Your immune system is kind of knocking it back and keeping it at bay. You can detect it in tissues by DNA and stuff, DNA analysis. It can be reactivated and actually get into the secondary infection, a more serious infection, after immunosuppression. So this happens to people that are organ transplants or chemotherapy, and so you can start to see this thing coming up. Not that many, but some will go into the secondary active infection, and then you end up having the symptoms of full-blown TB. So, a number of different ways this can go. Most folks, if they do end up getting it, are going to end up with a latent infection. A small subset of those guys, it's going to end up becoming a actual active infection. And for the most part, you have no disease at all. The number of diseases and things that's possible to get is staggering. And the fact that we're not all, sick and dying all the time, I like that. So TB is something that is a lot of different populations had different concentrations of TB. And it changes over time. With proper medical treatment, you can begin to suppress the amount that's in the population and reduce the spread of it pretty effectively. We see then that there are a lot of TB cases that are brought in from by people from other parts of the world, and so in other parts of the world, particularly in parts of the third world, TB is increasing. So a couple of things are interesting about TB. Like diphtheria, we could generate a vaccine against it. And it's an interesting kind of game in that in the U.S. we have no vaccination. So we don't prevent infection, but the infection is fairly low rate. What you can do in a population that has not been vaccinated is you can do the TB test. Because once you've been vaccinated, if the vaccine worked, you have an immune response to it. And the TB test won't work. Everybody is going to light up the TB test like a big dog. But if you're not vaccinated, you can do a TB test and identify people that have TB and say, okay, we need to put you, on a course of antibiotics, and you can't go work in the nursing home, you can't go work in the daycare center. And so, as it turns out, everybody that's going to go work at any kind of YMCA, YWCA, man, before they let you in the door, you got to have a TB test that was current within the past few months. And I would imagine nursing homes as well. So that's the surveillance trade-off that we've got here in the U.S., is that because we don't vaccinate, we can track it. In Europe, the BCG vaccination is a drug that is used, and that means that you can't track any active disease. And again, it's a kind of thing that you don't get herd immunity from this because the bacteria is present all the time in nature. So it's always going to be around, and if you haven't been vaccinated, you're at risk of being able to get it. So it's one of the ideas with a viral disease is if you vaccinate, 60, 70%, probably 70, 80% of the population, you can make it so there is no reservoir for that virus because the virus can't replicate outside of a host. Bacteria, they're replicating in the soil, they're replicating all over the place. It's not an issue for them, and so you are never going to actually deplete the amount of the actual disease-causing agent. So the direct observation treatment system is what our approach, and we can begin to actually have surveillance in the population, and this allows you to limit the exposure and to limit the number of people that are exposed. But it is an interesting kind of approach to it. Vaccinations versus no vaccinations. Drug resistance is actually a serious problem in that we've got these terms, MDR, which stands for multi-drug resistant. Multi-drug resistant bacteria are the real enemy of epidemiologists or doctors in general, because multi-drug resistant bacteria are resistant, as the name implies, to a whole range of different kinds of antibiotics. And sometimes this will result in a bacterium that cannot be killed with an antibiotic. All right, let's see what else we're going to go here in a moment. So, as we're talking about lower respiratory, not upper respiratory infections, we get into the area of pneumonia. And this is where we have the bacterial infection in the lungs. Pneumonia typically occurs because you've had some kind of immunosuppressing or immunostressing condition. And a standard thing you'll see is that when people get the flu, a week or so after their bout with the flu, they come down with pneumonia. And I'm sure the same thing is happening with COVID. And that's because the flu infection is stressing your immune system. Your immune system is not able to maintain its vigilance. And these bacteria that are present really in you all the time get a foothold. So there is atypical bacterial infection. And this is when you snort up enough bacteria that you actually overwhelm the system and you get the infection. But that's not, it is atypical. That's not the standard way to do it. So you end up having the sort of standard things you expect from a lung infection. Coughing, chest pain, blood in the sputum, a lot of things that start to indicate that you have a bacteria involved. Mucus begins to build up. You can also start to have damage in the tissues as well. So there are a range of bacteria that cause pneumonia. The Streptococcus pneumoniae is by far the most common. There are other bacteria that cause pneumonia. And this is just, again, one of these terms here, that pneumonia is a generic term. And we're talking now about some of the bacteria that can cause it. So Haemophilus does a moderate amount. Staph aureus, this is a bacterium that is present on the skin all the time. And when you heard about MRSA, methicillin-resistant Staph aureus, that's what we're talking about. It can get into the lungs and get a foothold. fairly dangerous because it actually begins to actually kill the tissue. Klebsiella is another one. It's also got a lot of antibiotic resistance, and so you don't have a whole lot of treatment options for that. Pseudomonas, we actually were looking at this one, and it's opportunistic in that only during a pretty weakened immune system do you actually end up with it. So the elderly end up suffering from it. The lung capacity is not as good. The heart is not able to push fluid in the lungs as well, and so you start to have problems as the lungs begin to fill up. We have these terms that are known as walking pneumonia, which is essentially a fairly mild case of it. And then you also have the lobular, which is only one lung. Nobody ever refers to that. But you may hear people talking about double pneumonia. And it's not that you've got like 2 cases of it. You have the bacterial infection in both lungs. All right. We're going to knock off here, and I'm going to go cough for a while. And we'll pick up on Friday. And today in lab at 1, we're going to do a range of different tests. Casein hydrolysis, looking for bacteria's abilities to secrete a protease to break down milk protein. We're going to be doing a gelatin assay, and that Jello is not like agar that we use for our agar plates. And I killed the video, but regardless. Jello has a small protein core with a big amount of sugar coming off of it. If you clip that protein core, you will degel Jello. And so there are some bacteria that secrete a protease that can degel Jello. And so we'll be looking at that and a couple other metabolic enzymes and things as well. So I will catch up with you at 1, and that's all we have to say.
Did you give out the lab email on Monday?
I did, and I think, did I post it to Moodle yet?
Oh, I don't know.
I might not have.
I can just get it from
Yeah, I mean, all you're going to do is going to be a group of bacteria, and you're going to inoculate either the gelatin slant or different solutions. It's not particularly, tomorrow we have some things we have to do. We have to add different reagents and things and observe. But today is pretty much just incubation.